There we go. Welcome. Today our speaker is Dr. Roland Lill, and he comes to us from the University of Marburg, Germany. I'm going to introduce him in just a minute, but I want to share my screen with you and remind you of the website that you can go to for the Centers of Excellence in Hematology. This is the homepage link where you can find here lots of upcoming seminars that are happening. You can find, especially, I want to draw your attention to this link right here, which announces funding opportunities that are available to anyone. That specifically is noting a funding opportunity for what are called Type A grants, which are essentially core credits, so that you can, at any of the centers across the country in the United States, you can actually apply for grant funding to support some use of the cores that are available there. And I want to just show you some of those cores quickly. So for example, at the Fred Hutch core, you can go in and find protocols of how they isolate CD34 cells, and you could write a grant if you need to use those.
Diane, can I interrupt you? We don't see anything.
You're not seeing my screen?
No, it stopped sharing somehow, right in the beginning.
It came off. Wow. Okay, let's try it again. I don't know what happened to that. Let's try.
In the beginning we saw it and then it stopped sharing.
Yeah, then it disappeared. So let's try it again. Are you seeing it now?
Yes,
you're
it's
back.
there.
Thank you. Thank you. Sorry. Please interrupt if that happens. This is the Center of Excellence in Hematology website. And I just want to show you a couple of features of it, which are the up and coming seminar series. And also importantly, that there are funding opportunities to use the cores that are available at these centers. And if you click on that link right there, you'll see announcements for funding opportunities. The very top one are Type A awards, where you can get credits to use different cores at these centers. Some of those cores include, if you click on the protocols, you can find cores at the Fred Hutch Cancer Center, cores at the University of Utah, which include an iron, heme core, et cetera. Cores at the University of Indiana and Yale. And I highly recommend you go look at those funding opportunities, because it's a great opportunity to learn what's offered at those cores, but also to utilize those cores when you might not have those services available at your local institute. And so if you have any questions about it, I recommend going to the homepage and you can kind of snoop through what the different opportunities are here. And you can also reach out to me if you need any help surfing through this information. So I'm going to stop sharing there. And now I want to introduce Dr. Lill to you. So Dr. Lill comes to us from Germany at the University of Marburg. He got his Ph.D. I'm going to just completely cream the German pronunciations, and I apologize for that. In biochemistry at the University of, is it München?
Munich.
Munich. See, I don't know. Okay, I know it's München. German,
it's München,
but that's okay.
in Germany.
At the University of Munich in biochemistry. He actually then went on to do a postdoctoral fellowship at UCLA, and then joined back to be a researcher at the University of Munich, and got a faculty position at Marburg. And how long have you been there, Roland?
24 years.
Congratulations, that's awesome. And so he's a full professor there. His research is focused on understanding iron-sulfur clusters and iron-sulfur cluster biosynthesis in the context of disease, and using lots of models and crystallography and all those things that you will learn more about today. The title of his talk is Biogenesis of Iron-Sulfur Proteins in Eukaryotes, Mitochondria, Mitosomes, Mechanisms, and Maladies. And I'm going to just remind you that if you have questions, to please populate them in the chat. And then we'll take opportunities to invite you to ask your question in the middle if it's appropriate, or we'll hold them to the end. But please feel free to speak up. At this time, I want to just invite you to please mute yourselves if you are not a speaker. And I can help do that if you're not able to. And so Roland, it's yours. Thank you so much for speaking today.
Yeah, thanks a lot. I think you can see my screen already.
Looks great.
You see not only Marburg on top, on the bottom you see the building of my institute, which this week, after 24 years, we leave. We go from the Lahn Valley. The Lahn is the river in Marburg, from the Lahn Valley to the Lahn Hills, some 2 miles away, into a new building where we join the Institute of Synthetic Microbiology. So it's really difficult to move from such a very nice building because the new building is a normal, modern building, but it's of course nice because we're again equipped with very nice instruments. So what I'm going to talk to you about is what we learned from iron-sulfur protein biogenesis, and basically almost all of my time in Marburg, I dedicated my scientific work on this problem, how iron-sulfur proteins are made. And I would like to start with a transparency where I show you which kind of iron-sulfur proteins in cells are present and what they actually do. And you will see many different processes are supported by iron-sulfur proteins. Very classical, you certainly know that conitase. Helmut Beinert has investigated this very intensely in the citric acid cycle. You know the complexes 1 to 3 of the respiratory chain, and also the ETF dehydrogenase for beta oxidation of fatty acids. Iron-sulfur clusters, iron-sulfur proteins are also involved in the biosynthesis of other cofactors, protein cofactors. For sure, you know ferrochelatase, at least in human cells, not in Saccharomyces cerevisiae, as a 2-iron-2-sulfur cluster. So basically, heme synthesis also depends on at least one iron-sulfur protein. And I want to mention molybdenum cofactor and lipoyl biosynthesis because I will come back to this in the later part of my presentation. In yeast, some usually essential amino acids in human are made in yeast, Saccharomyces cerevisiae, and they involve some iron-sulfur proteins for help. They have been lost in humans, and probably this is one reason why we have to eat these amino acids. Well, we have functions in the redox metabolism and most prominently in iron regulation. You all know IRP1, iron regulatory protein 1, which is involved in translational regulation of a handful of proteins. And translation is the target not only by the IRP, but there is this kind of famous protein RLI1 or ABCE1 in human cells, which splits the ADS ribosome after translation termination into the 2 pieces, 60S and 40S, to help reinitiation of the new translational process. Translation is also the target indirectly because many iron-sulfur proteins are functional in DNA modification, for instance, radical SEM proteins as shown here. And also the nucleotide metabolism itself, either degradation or the biosynthesis of the nucleotides requires iron-sulfur proteins, and they are mentioned here. Nucleotides play a role also in the nucleus because DNA synthesis in play of polymerases, DNA polymerases have an iron-sulfur cluster. If you delete the binding site of the iron-sulfur cluster, they cannot bind to accessory proteins, and this hampers DNA synthesis. So you see, DNA synthesis and genome maintenance is an essential topic where iron-sulfur proteins come into play, and this is also important for DNA repair. Various enzymes like helicases, ATP-dependent helicases, have an iron-sulfur cluster, and I can go on and on and on. Chromosome integrity, many proteins depend on an iron-sulfur cluster. Mitosis has a kinesin, we recently published with an Israeli group, and this is also dependent on an iron-sulfur cluster for function. Well, there are mitochondrial proteins or mitochondrial attached proteins, the neat proteins. They're also sometimes called cis-D, 1 and 2 and 3 proteins. It's not so clear what they are doing. And they also have an interesting 2-iron-2-sulfur cluster. Well, we come now to virology, where all very many very much influenced by virus attack at the moment. But we have, fortunately, a viperin protein and also a radical SEM protein, which produces a DD-hydroxy CTP. And that's integrated into the viral genome when viruses synthesize their genome again. And this is the first defense against viruses. But even viruses, as we recently learned, make use of iron-sulfur proteins, as Tracy Rowell's group published recently. The RNA polymerase of SARS-CoV-2 apparently has an iron-sulfur cluster. So you see, the whole world is full of these beautiful iron-sulfur clusters. And we are interested, as I said, since 25 years, how these clusters are made. And what we learned, and many other people learned, is that in mitochondria, the process starts. We need the so-called iron-sulfur cluster assembly machinery, which is very much alike a bacterial machinery. So it's evolutionarily inherited from bacteria to end up in mitochondria. And in the cytosol, we termed the machinery needed for nuclear and cytosolic proteins, the CIA machinery. Actually, I got the permit from Jerry Kaplan because I said, you know, what do you think about this name, CIA machinery? He said, go for it, it's better than our CIA. Just a little joke. And the two machineries actually are connected by an ABC transporter because the mitochondria export a component which is still not completely clear what it is. It possibly contains iron and sulfur and some other chelating molecule. And this is exported by this ABC transporter in a glutathione-dependent fashion, as you see in this model. Well, it's not so simple because more than 30 proteins are involved in this process, and you see them all summarized in the mechanism as we know it at the moment. And the process is highly conserved from yeast to human. So whether organism you study, it's basically almost the same in all of the eukaryotic organisms. And you see the names of the people who contributed to the discovery of many of these proteins. And I think we also discovered like almost more than 50% of these proteins and described them the first time. So today I will concentrate on two things only. I will tell you after a more broad introduction, I will tell you a little bit how 2-iron-2-sulfur clusters are assembled. That's the initial step of iron-sulfur cluster biogenesis for the entire cell, because you see, and I possibly have to use my laser pointer here, you see that basically the mechanism of the whole process, it starts here. Then the cluster, the 2-iron-2-sulfur cluster, is transferred, converted to a 4-iron-4-sulfur cluster. Then this famous XS molecule goes out, and then another machinery, the CIA machinery, in a sequential fashion synthesizes the cluster for the 4-iron-4-sulfur and 2-iron-2-sulfur proteins in the cytosol and nucleus. And the second thing I want to do is I will concentrate on the assembly of 4-iron-4-sulfur clusters in the end, just with a few slides. Well, let's start a little simpler. As you already know, we have the ISC assembly machinery, the CIA machinery, and they're connected by this ABC transporter. And this is actually, or many of these proteins, the green ones, they are essential proteins. So if they delete them in yeast or humans, then the cell dies. And this tells you already that also our process, the assembly process in the mitochondria and the cytosol, is essential. And most of the proteins, if you delete them, lead to lethality of the cell. Well, the process is not only essential, It's the minimal function of mitochondria, which I, as a biologist, find very intriguing and interesting. What do I mean with the minimal function of mitochondria? If you go to microsporidia, these are organisms which are relatively close to fungi. Two examples here, Encephalodozone and Trachypleistophora. Then these organisms do not have any classical mitochondria anymore. They have just the reduced mitosomes. So all the genes for classical mitochondria have been lost by reductive evolution. They do not even have heme biosynthesis, no respiratory chain functions, and all the things you know what mitochondria usually do. But these mitosomes still have a double membrane, and what they have, otherwise I wouldn't tell you, they have a ISC assembly machinery, and only just 8 proteins, the core ISC proteins, are left over. And we could show that they're actually functional, and apparently even mitosomes follow the same mechanism with a reduced capacity of proteins. What I depicted here, they have this ABC transporter, and on almost full CIA machinery, this is not so much reduced as the mitochondrial machinery to synthesize their proteins.
Hey, Roland.
So, yes.
This is Diane. Will you go back one slide?
Yeah, sure.
So are there species that are pretty close to that have a normal mitochondria? Where do these guys live?
These guys, they are fungi. I kicked out the evolutionary tree, unfortunately. They are very close to fungi in the evolutionary tree of eukaryotes, but you find mitosome-containing organisms throughout the complicated evolutionary tree. So in all 4 or 5 branches of the evolutionary tree. These microsporidia, they are usually pathogens of immunocompromised people. Usually they live in low animals like cats and mice and so on. And we don't, this is no danger for us unless we are immunocompromised. And some of these beasts can provide or can give you a problem, health problem.
Okay.
That's why they are also interesting in a way.
Yeah. Thanks.
Yeah, but they apparently have, I just looked it up, they do not have heme, they have just a cytochrome B5, what I found, so maybe, but this is not known, maybe they take up heme from the environment. They cannot synthesize it, so they have no genes for making heme. Okay, here's the mitochondrion again, and here I tell you, without going into detail, what the concept is of making, for instance, 2-iron 2-sulfur proteins or clusters, or 4-iron 4-sulfur clusters. Without going into detail, as I said, so there is a first reaction where we synthesize the 2-iron 2-sulfur cluster, and I will go into this later on. Then we transfer this iron-sulfur cluster by trafficking to a glutarredoxin. And from there, you can spontaneously make 2-iron 2-sulfur proteins, or then you can do another synthesis reaction by transferring this cluster, as you will see later on in my talk, to the ISCA proteins, and they give rise to reductive fusion of the cluster, as we recently found, to, for instance, give the cluster, the 4-iron 4-sulfur cluster for aconitase and many other proteins I mentioned to you already. So that's the general mechanism. And we need basically specialized proteins for the synthesis, trafficking, and insertion reactions. And we can nicely sort these proteins in vivo to find out where in the biosynthetic chain these proteins are localized.
I have a question.
Sure.
This is Caroline.
Yeah.
You have placed BOLA3 downstream of your apparatus that assembles 4-iron 4-sulfur clusters.
Yeah.
What do you think it
is
does?
a very simplified model.
What do you think it does?
We don't know. I mean, you know that the simplified model is also a little bit too much simplified because it also binds to the GRIX5. And in that sense, it transfers clusters, or that's at least the view, transfers clusters from GRIX5 to downstream proteins. But then the BOLA3 should be, if this is the true function, should be or should have a very similar phenotype if it's here as the IBA57 ISCA1, ISCA2 proteins. But if you look at the phenotype in vivo, it doesn't have it. And that's why this GRIX-BOLA3 interaction is probably not giving rise to the molecular phenotype in vivo. And that's why I place it here. And I don't really understand what the GRIX5 BOLA3 interaction actually means. That's absolutely clear. There's also a BOLA1, as you know, and that's probably not even involved in iron-sulfur cluster biogenesis. We have no idea what this protein is doing, but the phenotype is 0 for iron-sulfur cluster biogenesis, even though it's very, in vitro you can do transfer reactions, but in vivo you don't see it. And that's why my group is usually also looking, as you do, Carolyn, always looking into the in vivo function and then doing the biochemistry to see that our biochemistry fits with what a cell requires or tells us about these proteins. And if you only do in vitro stuff, you sometimes get fooled. Did this answer your question? I know not satisfactorily because we don't really know what the true function, biochemical function of BOLA3 is, but the phenotypical location is correct here.
I think I just have to be satisfied that we aren't done with all this work.
No, not at all. Don't worry. Well, the process, you might be interested in this, is very much connected or interwoven with human diseases. The most prominent one, the founding member for human mitochondrial diseases involved in iron-sulfur protein biogenesis, frataxin, giving rise to this neurodegenerative disease, Friedreich's ataxia, which is the most prominent, most prevalent ataxia, affects like 1 in 30,000 people. I usually say 3 people here in Marburg possibly statistically should have Friedreich's. So it's not that rare. All the other diseases are much rarer, much more rare, and they give rise to neurological, hematological, or metabolic phenotypes. For instance, the hematological phenotype is relatively easily explained by a defect in the ferrochelatase. I mentioned it already, and that would give rise to a lower heme synthesis if clusters are not made. What is also prominent is that often when you have a functional impairment of these early factors, the core ISE factors, that you get a mitochondrial iron accumulation in yeast and also in human cells. And this is connected to IRP1, as we think, or to the aft system in yeast. Because if iron-sulfur cluster biogenesis is not working, the yeast system and the human system, by completely different mechanisms, talks to the iron regulation system, and this brings in, as a result, more iron into the cell, and in this case into the mitochondria, and this gives you often some redox stress, which apparently also leads, at least in yeast, to the degradation of heme, and by a fashion, chemical fashion, nobody knows at the moment. Also, the late factors here giving rise to 4 and 4 sulfur clusters, they are summarized under these multiple mitochondrial deficiency syndromes. And the most prominent phenotype is that the lipoyl synthase gives the phenotype. And if this is, this needs 2 iron 2 sulfur clusters, and if you don't have a functional lipoyl synthase, you don't get lipoic acid, and you see the lower lipolation of PDH, pyruvate dehydrogenase, and alpha-ketoglucan dehydrogenase. That's a phenotype we and others described first, and that's now very much used in the clinic to diagnose people in such proteins and in the lipoyl synthase function. The mitochondrial iron in this case is normal because this bar here going to the iron regulatory system is independent of these proteins. So now I would like to come to my first data topic after this broad summary, which Diane asked me to do that for this audience. I will ask the question, so how is this actual synthesis of 2-iron-2-sulfur clusters in the mitochondrion accomplished? As you see, this needs a complex of six proteins. And the complex is even a dimeric form, as you will see. And this gives rise to a 2-iron-2-sulfur cluster. Again, this model is a very simplified form. So what do we know? Also in a very simplistic manner, what do we know about the synthesis process? We need a sulfur donor if we want to make an iron-sulfur cluster, and that's the sulfur donor is the cysteine desulfurase NIF-S, which teams up with two small proteins, the acyl carrier protein, actually discovered in Salt Lake City by Dennis Dean and Jared Rudder, and the small protein ISD11, which was discovered by Walter Neupert, Nikolaus Pfanner, and our group. And they stabilize the NIF-S as a sulfur donor. NIF-S generates this persulfide and SSH group, which later on is transferred to this scaffold protein, ISO1, on which the synthesis of the cluster actually occurs, as you will see in a minute. Then we need an iron donor, and there's a lot of discussion what the iron donor actually is. Recent data indicate that ISO itself brings the iron already along when it binds to the NIF-S ISD11 ACP complex. But earlier data suggested that the frataxin is actually an iron donor. Later experiments, mainly from the group of Barandeau, Dave Barandeau, have shown that the transfer reaction, which you just saw, where the pulse sulfide is transferred from NIF-S to ISO, that this is supported by a conformational change or inducing a conformational change. This is the function of frataxin. Maybe both is correct, but it's of course clear that the ISO brings along the iron. What then happens is we need, as we initially showed in 2014 already, we need a ferredoxin, we need electrons, because the sulfur has to be reduced to sulfide, and then we have a sulfide intermediate, which is not shown here, and an iron intermediate. And then the next step, giving rise to this dimeric form of ISO carrying a 2-iron-2-sulfur cluster, is very poorly characterized. And this, I will concentrate on this step now. What I will tell you is that the dimerization of this holo iso dimer, that this is induced by two N-terminal tyrosines, which are highly conserved in nature, universally conserved, I have to say. And we recently found out what these tyrosines are actually doing. I have to introduce you now to a reaction which we created in 2014 using CD spectroscopy, initially used by, for instance, Mike Johnson in the bacterial system, and Bonomi, who introduced this very nice assay. And we applied it to mitochondrial proteins. And as you see, if we have the complete system of all these proteins, here shown here, then we get the cluster synthesis indicating by a change in the CD signal at 431 nanometers. If we leave out one of these components, just one, then the reaction dies. If we leave out cysteine or iron or NADH, or an ADPH, which is needed for the ferredoxin reductase to reduce the ferredoxin again, then the reaction dies and we go back to a background reaction. So apparently we can monitor the 2-iron-2-sulfur cluster biogenesis by this system. And another important point was the crystal structure, which was done by Marek Boniecki and Mirek Ziegler in Canada, and we collaborated with them. You see here the NIFS very nicely intertwined. Here is a cartoon to orient you a little bit better. Here's the ISD11, here's the ACP, and the ISO binds at the tip of this dimer of the ISQ. You see the pyridoxal phosphate. The NIFS is a pyridoxal phosphate dependent enzyme and releases the sulfur from free cysteine onto a flexible arm to create this persulfide, as you will see in a minute. And then the cluster is synthesized here, where you see a zinc bound in this crystal structure, which is of course an artifact, an in vitro artifact. Here normally the iron would bind and the cluster would be formed. And if you rotate this a little bit, then you can probably see it a little bit better. You also see that the ISD11, which are these 3 helical bundles here, have a fatty acyl chain of the ACP, of the acyl carrier protein, attached via the phosphopantenic acid. And this is kind of a sticky finger going into these helical bundles, and the cluster is actually synthesized here.
Roland, we have a question in the chat. I just want to invite Annalise to go ahead and ask it.
Yeah. Can I read it?
I can read it for you.
Oh, yeah.
It says, what is known about the regulation of the mitochondrial iron-sulfur cluster assembly? I don't know if she wants to elaborate more on what that, more specifically what she's asking.
Yeah, the regulation is on various levels. It's of course regulated by the iron availability, and the cell delivers the iron to the mitochondria, and I think iron limits the synthesis activity. Then a second level of regulation is the stability or the degradation, I should possibly say, of the ISQ protein, the ISQ scaffold protein. Possibly, and I'm not 100% sure whether this reflects the data completely, if the ISQ protein is not actively involved in the process, not binding to the NIFS in its apo a long time, then it's degraded by the possibly by the LON protease or a major mitochondrial protease. These are the 2 major levels of regulation.
Thank you so much.
Thanks, sure. Okay, here several crystal structures and also cryo-EM structures have been used to produce this model or this movie by Sven Freibert. You see the site close to pyridoxal phosphate where this persulfide, indicated by this yellow square or ball here, is made, and then this loop here swings out to the active site where the zinc binds, where the cluster synthesis actually occurs. Well, this is of course computer gimmicks, but in a way, in a similar fashion, the mechanism might work. Okay, here's again 3D view, or it's only in 2D for you, of course. Space filling, and here with secondary structures where you nicely see where the cluster is actually synthesized. I can highlight it for you, which is here. And this also shows us a very nice feature that the ferredoxin binds according to our Sachs structures. There is no crystal structure yet or cryo-EM structure for the ferredoxin binding. But in this Sachs data, we find the ferredoxin about 14 angstroms away from the reactive center. So this is a compatible distance for electron transfer as we know it from the respiratory chain And so it makes sense how we placed the frataxin in a way. And you see the cartoon here, how this actually works. And I would like to mention again, whenever I show you the NIF-S in a collaboration or in interaction with these other proteins, I usually show only 1/2 to make it less complicated. And here's the mechanism. Cysteine is converted to a sulfur. Iron comes in then by some magic mechanism using electrons, reducing the sulfur. The iron-sulfur cluster is formed. And we were interested to get a little bit more insight into this, and we became interested into this N-terminal tyrosine of isQ protein. And I think we have a nice function for this now. So in the sequence alignment here of the isQ protein, you see that all proteins actually start or have an N-terminal very close to the N-terminus tyrosine. There is virtually no exception. All proteins have it. The only exception we know is the isQ1 in the cytosol, but we can show that this is not a functional protein because it lacks the tyrosine. I don't go into this today. We could show already in 2004 and Takahashi in 2019 in E. coli that this tyrosine, this conserved tyrosine, no surprise, is essential both in yeast and in bacteria. And we were interested whether this is also true in humans. And we constructed mutants, converted the Y, the tyrosine, to an A, or we also deleted the mitochondrial localization signal as a control in a way. And as you see, when we deplete the isQ protein by siRNA technology, we, for instance, after 3 or 6 days of RNAi treatment, maybe we look only at the 6 days data, we lose mitochondrial iron-sulfur cluster assembly as seen in the activity of mitochondrial aconitase or succinate dehydrogenase as 2 examples. And the effect is relatively dramatic. We can rescue this by reintroducing into the cell the isQ2, but neither cytosolic protein, because we steal away the mitochondrial localization signal, nor the tyrosine mutation, which is shown here, can rescue this phenotype. So apparently also in human cells, this tyrosine is absolutely essential. It is also essential in the cytosol, not only in mitochondria. And this is also true for the protein. If you localize isQ protein into the cytosol without a mitochondrial localization signal, then you don't get any iron-sulfur cluster assembly, neither in mitochondria nor in the cytosol, even though, as I said, it was claimed that the isQ1 protein should be there. So this is not functional, apparently. And you can not only test cytosolic aconitase or the hollow form of IRP1, as you know, but also other iron-sulfur proteins, and they lose their stability because they're apoproteins and they're degraded. And this is an indirect, simple measure of iron-sulfur cluster biogenesis defects. So what is going on? What is this Y35, tyrosine 35 in human isQ2 doing? When we use our CD assay, which I showed to you already, here's the wild type protein. When we use the Y35A protein or an L mutation, whatever mutation we do, we fall back to a background as the same as we leave out frataxin. So also in the in vitro assay, generating 2 iron-2 sulfur clusters, we see what we see in vivo. It's absolutely essential. But the funny thing is, if we purify these proteins from E. coli and chemically reconstitute them, especially the Y35A, we get a reconstitution of the 2 iron-2 sulfur cluster almost the same way as in wild type cells. So in principle, this protein can make or can receive the iron-sulfur cluster, but apparently not in the physiological way, only in the chemical way. So this gave us a good indication that the protein is not simply dead. It's functional in a way to assemble a cluster, but it cannot do it on the normal physiological way. And then we used an assay showing whether the persulfide has actually arrived on the ISU from the NIF-S persulfide donor. In short, this is a modification with an MBP substrate. This has a little bit higher molecular weight, and running these things out on gel, you see that if you have a persulfide and cleave it by reduction, then you fall back to a lower molecular mass. And if you compare then the wild type protein with the tyrosine 35A protein, you see the efficiency of persulfide transfer in the two reactions studied in parallel are the same. So apparently all the steps before persulfide arrival at the isQ protein must be okay. Apparently, and obviously the function of the tyrosine 35 is in a late step and not in an early step. What could this late step be? Well, this could be I mentioned this already, the isQ2 dimerization, the actual iron-sulfur cluster formation, or also the dissociation from the entire complex, or all of these, of course. Well, we took us basically several years to understand what the true function is. Eventually, the important thing was that we asked our question, does pi stacking of the two tyrosines in two different isQ proteins trigger the dimerization? I sometimes call it a greasy handshake. Do these proteins make a greasy handshake? Which then would lead to iron-to-sulfur cluster. And this would be an elegant mechanism, because if you remember what I told you in the beginning is that each isQ protein would have a 1 iron, 1 sulfur intermediate. And if you combine this 1 iron to 1 sulfur twice, then you have a dimer on with the 2 iron, 2 sulfur. So that would be a nice mechanism. But how to show it? The interesting experiment was that we said, okay, can we replace the tyrosine by opposite charges and convert this greasy handshake into an electric handshake and mix, for instance, the Y35D mutant with the Y35K mutant? And I wouldn't tell you if this wouldn't have worked. We first looked whether the sulfur transfer still works. So as you see here, the persulfide transfer is very much the same in the mutant and in these two mutant proteins. So they're kind of active in the early reaction. But apparently in our full reaction, making the 2 iron 2 sulfur clusters, these proteins individually are completely dead. They cannot replace the protein. They are even worse than if you leave out frataxin. But the nice thing, if you now mix them in a 50-50 orientation, either to give the same concentration together as we have as a wild type protein, then you end up at a roughly 50% efficiency of cluster formation. If you then add 100 micromolar of each of these charged proteins, then you get the same efficiency of cluster synthesis. We still don't understand why the rate is a little slower and not the same, but there's certainly something behind this. So we were very happy to see this because we now apparently have a function of this tyrosine residue in the dimerization. Is it really a dimer which is formed by these mutants? We looked in gel filtration to separate the dimer and the monomer. And what you nicely see, if you record the protein at 280, you can nicely separate them by gel filtration. And the absorbencies at 320 and 420, these are the wavelengths where the cluster is seen. you see that only the wild-type protein gives rise to, and only the dimer gives rise to cluster formation. There is no signal whatsoever at the monomeric form. And also the mixture of the 35D and K mutants, when you mix them together, give rise to a nice dimer formation with cluster. So apparently we're right that the tyrosine is important for cluster formation on a dimeric
We have a question
form.
in the chat
Sure.
from
Yeah.
Leju. Matthew, do you want to go ahead and ask your question?
Sure, yeah. So
does tyrosine to phenylalanine recapitulate a cluster formation? We haven't done phenylalanine. Dr. Takahashi in E. coli has done it, and this gives activity again, and not as high activity, but also a good activity in E. coli. So it's a very good question. Yeah, so which is probably telling you that also the tyrosine or the hydroxyl group of this residue is important. You can as well replace it by two charges. In nature, this of course wouldn't happen because you would have to multiply your gene. You should make a gene dimerization basically, and then you have to change the 35 into different directions. This possibly doesn't happen so easily. Well, we then asked ourselves, if these tyrosines are close to each other, we should be able to cross-link. In the literature, I knew this from my PhD time. In the literature, you can nicely cross-link tyrosine residues here, as indicated by the German group, done with synuclein. And I gave this paper to Sven Freibert and asked him to do this experiment. And sure enough, and now here we go. Sure enough, we saw that we can nicely cross-link the ISQ protein in this reaction and give rise to a very similar spectrum as in the literature. And when we run out these, and as a control, of course, if we do this with the Y35D or K proteins, this doesn't work at all. If we then look at the dimerization on denaturing gels, then we see a very high efficiency of cross-link, indicating that apparently these two tyrosines can be cross-linked and these are the two protein or the two tyrosines that are cross-linked because the protein has more than just this N-terminal tyrosine, but they are usually inside the protein and cannot give rise to artificial cross-linking reactions. So with this, we come to a model, and I include data from other people for the whole cycle of 2-iron-2-sulfur cluster biogenesis. As you know, we start with cysteine, create a persulfide on the NIF-S complex, and the complex is shown here as a dimeric form. Then we load iron onto ISU, or ISQ1, with this N-terminal tyrosine, and bind it to the NIA complex. NIA stands for NFS IST11 ACP. Then frataxin comes in, and most likely its real function is to stimulate the persulfide transfer from the NIF-S to the ISQ protein. As I said, maybe there is also an iron-dependent function, but most likely not in the synthesis reaction. Then ferredoxin, our ferredoxin electron donor, reduces the sulfide, the persulfide to sulfide. Then we have this complex here. And now the scenario is that another complex comes in, which has undergone the very same reaction as this part. And then the two tyrosines are very close to each other on this complex. Each ISQ protein has a sulfide and has an iron. And if you combine those two guys, you get a tyrosine triggered dimerization by this greasy handshake, and you should get this protein with the 2-iron-2-sulfur cluster, which can be then delivered to the GRIX5. I mentioned this already. And then the components of these two complexes can be recycled and regenerated. For instance, you have to reduce the ferredoxin by the ferredoxin reductase. Okay, you may say, well, why should two full complexes meet? Why don't you do the 2-iron-2-sulfur cluster biogenesis on one complex, and that's sufficient? And then you have to do this in a way differently, because you could bring in an apoprotein from outside and bind here. If you have a 2-iron-2-sulfur here, as I told you, we never saw a 2-iron-2-sulfur on such a complex in our in vitro reactions. If this is the case, that's certainly an alternative. What would you get if you do it as I want, as I suggested? Make a tip-to-tip interaction of two identical complexes, you would get an iso-iso dimer. And if this is, for instance, his tagged, then you would get a dimer which carries two his tags. If you bring in a protein which has no tag from outside, if you give this in excess to these complexes, then you should get a mixed situation where one protein has no tag and the other has the tag. And this was our experimental approach. We did the reaction, separated the dimer and the monomer and the NIA complex after the reaction, and ran this out on gels. And as you nicely see, we can separate the ISQ his tag and the ISQ without a tag. It's here and there. And what you see in the dimer is exclusively, basically 100%, the tagged protein. And in the monomer, there is a little smear, but these are the ferredoxins and the frataxin. And when we do Western blot, for instance, we even more clearly confirm that there is virtually no his-tagged protein here. And the non-tagged protein, which we added basically together with cysteine to start the reaction, is completely in the monomeric fraction. And in addition, since the position of the ISQ his tagged is freed now, it also binds, because it was given in excess, it also binds to the NIA complex after the reaction. Before, it's not there, so you cannot see it. So I think this very nice experiment, which worked very efficiently, much more efficiently than I initially hoped when I suggested this experiment to Sven. I think convinces me that kind of a tip-to-tip interaction of this supercomplex gives rise to the reaction, and that this model seems to be at least suggestive to say this. With this, I would like to come to the next thing, and this is only like 3 slides because I think my time is running up, and I would like to tell you about the 4-iron-4-sulfur cluster synthesis. And as you will see, this is completely different and also resembles the reaction of the first part of the mitochondrial pathway just a little bit. So as you see in vivo, we showed, and other people showed this, what is needed for 4-iron-4-sulfur cluster formation is the donor, the supposed donor of glutaredoxin, which holds this intermediate 2-iron-2-sulfur clusters. It needs the 2 ISCA proteins and the IBA57 protein. I don't go into these proteins, what they actually do, or how they actually do it and what their structure is, but the phenotype of deleting these guys here is very much the same. So, for instance, you can measure this very nicely, recording the aconitase biogenesis. And we set up to analyze this assay, or this pathway. And in vitro, people have done this before us and have shown that from GRIX5, you can transfer the cluster to aconitase, and you get a 4-iron-4-sulfur cluster, and you just don't need anything, but this reaction requires the artificial reductant DTT. And so, of course, this is an artificial reaction. We know from in vivo, we need the ISCA proteins, the IBA57 proteins. And if we then mix all these components with an apoaconitase and measure then as a result of regeneration of the activity, formation of the cluster, measure the aconitase activity, yeah, we get a little bit of an activity if we add these three proteins. But we were not satisfied because we knew this activity is lousy. And so we knew already, or we felt already, that we need electrons. So we added the electron transfer chain. I already mentioned this in the beginning. It's also needed in the first part of my pathway. And when we now added the complete set, also these two components with NADPH, we get a roughly 50-fold stimulation of the reaction. And it's relatively fast to do this. When we leave out now the IBA57 from this complete system, the ISCA proteins from this complete system, or the glutaredoxin 5 as a donor, we fall back to this actually background aconitase activity regeneration. So apparently we can now with this system see that we have reconstituted the physiological process because we depend on all these proteins in vivo as well. And then we asked ourselves a funny question, because in human cells, not in yeast, there is a second ferredoxin, the ferredoxin 1. You know this under adrenodoxin. It is important for steroid biogenesis. So many steroids made in mitochondria depend on ferredoxin 1 as the adrenodoxin. But if we add this protein, which we could show is functional in vitro in steroid biogenesis, so it's not a dead protein, what we use here, but then we see no activity of ferredoxin 1 whatsoever. We can use other ferredoxins from, for instance, from this fungus Chetomium thermophilum. This works apparently, so this is a dual function of the protein. We cannot use thyroredoxins, for instance, and we cannot simply use artificial reagents, ascorbate and iron as reductants. We need specifically the ferredoxin to do this reaction. Well, we then basically said, how does this actually work, this 2 iron 2 sulfur cluster fusion to the 4 iron 4 sulfur cluster? Can we record this directly, even independently of the GRIX5 donor? What we generated is a dimeric situation of ISCA1, ISCA2, they form a dimer. And apparently each protein, according to UV-Vis and Mössbauer studies, has bound a 2 iron 2 sulfur cluster. And then when we ask whether this redox chain catalyzed by the ferredoxin reductase and the ferredoxin can transfer the electrons to get a fusion of these 2 iron 2 sulfur clusters to a 4 iron 4 sulfur cluster in vitro. And this worked very nicely. We can record, for instance, the oxidation of the NADPH to NADP at the 340 peak, or we can see this, the peak at 600, which is characteristic to 2 iron 2 sulfur proteins disappearing from here to there. Here you see it a little bit more nicely, because the 2 iron 2 sulfur spectrum is converted to a 4 iron 4 sulfur spectrum, which only has a peak at 420 or so nanometers. And you see this in a differential spectrum. If you make the differential spectrum early minus late, then you see a 2 iron 2 sulfur cluster spectrum, which is then converted to a 4 iron 4 sulfur. And when we then look directly at the rates, we are not absolutely happy with the rates because the reaction is relatively slow. We see that it works if we have the full reaction. If we leave out anything, it doesn't work. And the reaction is relatively stable if we let it starve for a while without ferredoxin, and only after, say, 45 minutes add the ferredoxin, then the reaction starts and actually very nicely in a fast reaction. So with this, We think we have reconstituted this reaction in a relatively physiological fashion. The model, which I showed you in the beginning, is simplistic, it's incomplete, because in addition to the IBA57, ISCA1, ISCA2 proteins, you additionally need the electron transfer chain, and we can show it in vitro now. In vivo, you cannot show it, because if you deplete ferredoxin or ferredoxin reductase, you know that we need it already here. All the later steps are compromised, and then you, of course, do not see another reaction, another requirement in this reaction here. So the ISCA proteins apparently have two functions. We see it as a scaffold for the 4-iron-4-sulfur cluster biogenesis from a 2-iron-2-sulfur intermediate. And of course, they are kind of trafficking proteins in the way that they shuttle or receive 2-iron-2-sulfurs and shuttle them on to targeting factors as 4-iron-4-sulfur clusters, which then in a protein target dependent fashion deliver these clusters into various iron-sulfur proteins, which I haven't talked about. Yeah, with this, I thank you for listening, staying with me. My thank goes to my coworkers. This is still my old institute here. And you see the name Behring. The Behring company, I should mention this, is producing now the vaccine, the RNA vaccine for BioNTech here in Marburg, and they predict that they produce here in Marburg 1 billion per year of vaccines. So we are pretty happy because almost 60% of the Germans are vaccinated, and this is due to the Marburg Behring companies. I would like to thank also my collaborators. I mentioned Michal and Mirek, and Dennis was also part of this project because he spent some 4 sabbatical months 2 years ago in our institute, and we had a lot of fun together. And I would also like to thank the funding from various different sources. Thank you very much for your attention, and I'm happy to receive questions.
Thanks, Roland. That was great. I think, Amit, do you want to unmute yourself?
Yeah, hi, Roland. This is Amit. Great talk.
Thanks a lot.
I was just curious about your sense of which are the rate limiting steps of iron sulfur assembly, whether it's that cysteine desulfurization or the transfer process you were describing. And sort of a related question to that is, if you remove iron sulfur cluster acceptors, what happens to synthesis?
Mm-hmm. You mean in vivo or in vitro?
In vivo. In
vivo.
Yeah.
So if you deplete, for instance, a conitase or so? Is that your question?
Yeah, if you depleted the complex 1 or 2 or a conitase, where do you see stalling if it occurs?
Yeah, I'm not sure whether this has been studied so well. Let me first go to the first part of your question. What is the rate limiting step? It's certainly not the desulfurase reaction, because that's a very fast reaction. Dave Barrandau has studied this. This is in the second range or sub-second range, how this persulfide is formed. It takes a little longer to transfer the persulfide, but this is also a reaction which is finished in 10-15 seconds. So it's a very fast reaction. As you have seen, the actual 2-iron-2-sulfur cluster formation takes a little longer. And this is probably due to what I call crazy handshake. The complexes have to meet and hit each other to actually form the dimeric form. I think this might be the rate limiting step, but we haven't, let's say, chased the actual rate limiting step in more detail. It's at least the slower step. The second part of your question, what happens if you remove target proteins? Well, in yeast, for instance, a major receiver is the cytosolic protein Lu1. You may remember this protein. That's how we started in the cytosol. It's an isopropyl malate hydratase or isomerase. And this is a major protein. And if you remove that, what you usually see is that the maturation of other proteins, other cytosolic or nuclear iron-sulfur proteins, is in a way better. Yeah. So if you add leucine to the medium, they don't need Lu1, but how the regulatory reactions occur, actually, this has not been fully studied yet. We're still busy with, let's say, the assembly processes and look into these various reactions. For instance, this is also an important reaction, which I skipped because other people are analyzing this reaction, the chaperone function.
Yeah, in the literature, there's sort of a smattering of reports of PTMs on some of these factors. And I've always been curious about which of these may be relevant in controlling, rate determining steps. But it sounds like from your response, a lot of these are happening pretty quickly anyway. So the nature of these PTMs or their significance, may, it's hard to think about.
Yeah, no, this is well taken. My take of these PTMs is that this is kind of not a major regulatory system. This is more kind of a fine tuning of the reaction.
Thank you.
I believe, if you look at, let's say, only one modification, this might not give rise to a major regulatory effect. But if you combine them, then this may give you more. And this is, of course, experimentally difficult to study. Yeah.
Thank you.
I think these modifications do something, but they are certainly not major effects on the system.
Caroline, do you want to go ahead and ask yours?
So, Roland, I'm looking at your cartoon and I'm wondering, have you examined what components of this machinery might directly interact with ABCB7?
Yes, we did.
And whether that tells you anything about what the potential substrate for ABCB7 might be?
I turned the thing around. I tell you which components interacts with ABCB7 and you tell me what it means.
Okay.
As you might imagine, we see an interaction of GRIX5 with ABCB7.
Okay.
So it's not unlikely that, let's say, there is a direct interaction. I'm sometimes a little bit skeptical whether this is true. But Sven also has used modeling of the structures, and you can model the interaction. But I think you can model many interactions by using these tools. But this certainly, by IPs can be seen in vitro, or taking materials from live cells, or also by purified proteins.
So,
as you know, we have the structure of ABCB7. We have now cryo-EM structure in collaboration with the Frankfurt group, which is in the closed state. And so we have a very nice situation. We have done HDX on the ABCB7, where we see the movement of these things. So we have a few things to publish on the ABCB7.
So do you think that it's actually transporting a 2-iron, 2-sulfur cluster?
I would say it's not excluded. It would fit to what let's say, all the physiology, the receiving physiology in the cytosol tells us. So it would certainly fit. It would not explain to you how this cluster is stabilized. You know that Jimmy Cowan has the idea that it's a tetraglutathione stabilized 2-iron 2-sulfur cluster. I have difficulties with this model because the glutathione concentration in the cell, also in mitochondria, is 10 millimolar. And if you cover cluster with 4 glutathiones, then of course what ABCB7 in its cavity sees between the 2 dimeric parts of the ABCB7, what it sees is glutathione. And of course you compete then with the 10 millimolar glutathione, and I'm sure your 2-iron-2-sulfur cluster chelated with the glutathione is at best in micromolar concentration, so it's much lower. And then I see a competition, and this would mean that ABCB7 mostly pumps glutathione, and that would be a futile cycle, and that doesn't make sense. So in my way, so I think my gut feeling tells me that glutathione certainly is part of this complex, which I call XS, but there must be something else. A more specific small molecule which helps chelating and stabilizing the 2-iron-2-sulfur cluster. That's the best answer I can give you. And you know that I sometimes gave up in speculating about this because identification of this XS escaped anyone in a way. Yeah.
We have one more question in the chat from Daniel. Daniel, do you want to go ahead and ask your question?
Sure thing. Thank you. Dr. Lill, wonderful talk. I just had a quick question. So the way that you have this cartoon described here, it looks like GLRX5 is the kind of deciding factor here. So where the iron-sulfur clusters are going to go. And I was wondering, if you knock down, say ABCB7, do you see increases in aconitase activity and mitochondrial aconitase activity? Or when you knock down ISCA1, ISCA2, IBA57, do you see changes to the CIA system in the cytoplasm?
Yeah, I see your point. Basically shutting off here, then the supply here is much better, and the mitochondria are happy. The problem is that ABCB7 has a major phenotype in the cytosol, and this strikes back to the mitochondria. I already told you that ABCB7 and these proteins compromised cells, they fill the mitochondria with iron. And this extra, probably non-chelated iron gives rise to an oxidative stress situation. I mentioned already that in yeast, for instance, we have shown this. It's probably the very same in human cells. There is a heme degradation, an active degradation by some chemical mechanism. And this already tells you that your expectation, which is logic, absolutely logic, is probably not fulfilled because other bad instances, other bad reactions prevent from making this more efficient, the mitochondrial assembly process. Yeah. And I think if you remember the plethora of cytosolic iron-sulfur proteins, which are not matured because this B7 is not working, this of course influences also the mitochondria in very large, and therefore I think it's kind of a social situation. The mitochondria produce something for the cytosol, hoping that the cytosol doesn't do bad things to the mitochondria in a back strike in a way.
Thank you. So we all have to get along.
Yeah, thanks a lot. Nice question.
Great. I think we'll stop there. If you have further questions for Dr. Lill, I'm sure he would be willing to answer them if you reach out to him through an email. And I want to thank everybody for their attendance, and hope everybody will be well. And keep going with your research. This is great fun to see everybody. Invite you to continue to look at the Centers of Excellence in Hematology website for information about new cores and tools that you might be able to use in your research. And thank you again, Roland. It was a great talk. Really appreciate it.
Thanks a lot.
Thank
Good
you,
night.
Roland. It's good to see you.
Nice to see
See
everybody.
you soon, Carolyn. See you soon, everybody. Ciao.
Bye, Diane.
See ya.